I've got my crazy orange gloves for you today. Today we're making osmometers. Osmometers are tools that can demonstrate water going up a tube by the power of osmosis. And we're going to do that with these things that you see on the table here. I have two solutions, a 30% syrup solution and a 10% syrup solution. And we're going to use those to create hypertonic sides of a membrane, and we're going to see osmosis occur. We also need some disposable pipettes. You can see the ends have been cut off as illustrated in your lab manual. There will be some changes to this lab as I demonstrate it. Here's our dialysis tubing. We know about that. I've got some pieces already in a beaker, and they're soaking. We also need food coloring. This is the Karo syrup that we're going to be using for our solutions there. It's just a commercial syrup. And then we have our beakers set up for our osmometers, one at 27 degrees, which is room temperature, and one at 90 degrees, which will be our higher temperature, not quite to boiling. And we also need another beaker for rinsing off our osmometers. And we're about ready. Okay, what do we need to do first? We need to make our solutions a certain color so that we can tell them apart. And we'll just pick out a couple colors here. We've got green. We'll add that to our 10% syrup, and that'll make it easy to tell apart while we're doing our little experiment today. Okay, so 10% syrup, we'll swirl that around in there. And for our other one, let's just go with blue. Add a few drops to our 30% syrup solution and stir that in a little bit. Alright, so now we've got those ready. Make sure they're mixed in real well. The syrup is kind of thick, so we need to make sure it's mixed. Alright, now we're ready to make our dialysis tubing osmometer. And this is a little tricky, and it's a little different than your lab manual. We're going to take our 3 mil disposable pipette. We've already cut off the end of the bulb, and we're going to slide that into the dialysis tubing and kind of slide it all the way in until it pokes out the other end. And again, that dialysis tubing has a tendency to stick together. And we have to do this carefully so we don't poke a hole through the tubing. Alright, it looks like we're about through. And we need to slide it on up over the bulb end as well. If the dialysis tubing gets dry, it gets a little harder to manipulate. Alright, there we go. Alright, you can see we've got the tubing sticking out past the bulb in both directions, actually. What we need to do now is seal it off by tying a string as tight as we can around the base of the bulb of the pipette and the tubing. So we're just going to lay it on the string, tie a nice tight knot, and it is important that it be really tight so that there can't be any leaks at that knot. Okay, we're going to tie it once, tie it again, nice and tight. Alright, and now at the other end we're going to fill that up with our solution. And this is pretty straightforward. We're just going to use our disposable pipette, and we're going to get 8 milliliters. So that's going to be up to the 3 milliliter mark 2 times, and up to the 2 milliliter mark 1 time. So we'll extract the 10% green 10% solution up to the 3 ml mark. And we'll squirt that down into the bag and also into the pipette, but we need to cover the tip so it doesn't drip out the other end of the pipette. If we don't cover the tip, it will just start to leak. That's an easy thing to forget to do as you're setting up this experiment. Okay, and there's another 3 mils. Okay, and now for 2 more milliliters, right there. Okay, now while we're still covering the tip, we need to clamp the top of that dialysis tubing, but we're going to fold it over to ensure a really tight seal, because this is one of the situations where if we don't seal it really well, it's going to mess up our experiment. We've got it folded over. And I'm going to put the clamp on just right above the top of the pipette bulb, just right above the bulb. We'll see if we can get that nice and tight. There we go. The fold is above the clip. And make sure it snaps together nice and snug. Now we can turn it over and remove our finger. Oh, and we lost a couple of drops there, but not too bad. We'll see if we can do it without having any drips next time. Okay, that's our osmometer number 1. We're going to make 4 of these. 2 with a 10% syrup solution and 2 with a 30% syrup solution. Okay, and you can see what it looks like there. You can see that there's a little bit of the green solution up in the shaft. What we're going to be looking for is a change in the level of that liquid. I'm going to leave that there. We're going to make 3 more, and when I get those made, we'll be right back. I've made the other osmometers. You can see that they look pretty similar. I have 2 blue ones with the 30% syrup solution, and I have 2 green ones with the 10%. And even though the level of the liquid in the shaft of the pipettes is a little bit different, that's really not too big a deal. Alright, next we need to rinse them off, make sure there's nothing on the outside of the pipettes. And then we're going to mark the level of the liquid. Let's go ahead and rinse these off first though. So I'm just spraying them off, make sure there's no extra liquid on the outside. Okay, and we're going to set 1 of each color in the 27°C beaker and 1 of each color in the 90°C beaker. Okay, so that one goes in there. Rinse off the blue ones. And we're ready. Okay, now we need to mark where the starting point is on each of these pipette shafts. And so I'm just going to make a little mark with a permanent marker. And we're going to see if that level changes. Now you might be thinking, how can that level change? We don't have any way of pumping water into that tube. But what we're looking for here is osmosis occurring, and an osmometer measures, meter means measure, so osmometer is a tool for measuring the effect of osmosis. And osmosis causes osmotic pressure, which can actually push liquid into the bag and ultimately up the tube. And we're going to let these sit for a little while and see what happens. Okay, it hasn't been quite an hour, but you can see that my blue pipette is so full now that it's almost ready to have liquid come out of the tip. So we need to go ahead and stop it. It's been about 50 minutes. And we're going to mark the change that we can observe in the level of the liquid on each of the shafts of our osmometers. Okay, so I've just marked that. You can now see that the level of the liquid has gone up a little bit. And we're going to measure how far each of those have gone. Okay, and these are our two from the 27-degree beaker. We'll mark that one as well. It's gone up a little bit. Okay, and now for the 90-degree. Okay, you can see where the level is. And for our fourth one, that one went almost all the way to the tip and out the top, and it would have gone out of the top if we had waited much longer. Okay, we can see all of those. Alright, so at this point we need to measure each of these, and again, we're not really interested in specific volumes, we're just interested in how far the liquid has moved up the tube, and it's just a relative comparison between them. So I'm just going to measure these, and if we lose a few drops at this point, it doesn't matter. We've already labeled the pipette shafts, so we really just need to measure the distance between the two marks. The first one appears to be 4.8 centimeters. 4.8 centimeters. Okay, this next one, this was in the 90-degree Celsius beaker as well. That one was 2.6 centimeters. So we'll keep both of these over here. Let me make sure, 2.6 centimeters for the green 10% syrup solution in 90 degrees. Alright, now for our 30% syrup, it looks like that's 1.3 centimeters. 1.3. And our last one is 1.0 centimeters. For a 10% syrup in 27-degree water. Alright, so at this point you're ready to draw some conclusions. So you need to think about which solution caused more osmosis, and also which temperature caused more osmosis. So draw your conclusions and write up your conclusions in your lab manual. And that's it.